If landing on the moon was humanity's grand voyage outward, then deciphering the structure of DNA was our most profound journey inward. In the early 20th century, scientists knew the secret to life. The very code of heredity was hidden somewhere inside the nucleus of our cells. They just didn't know what it looked like. That all changed in 1953, but the story of how it happened is a dramatic tale of ambition, rivalry, and a deep injustice. It begins in London in 1951, late at night in a King's College laboratory. A young scientist named Rosalind Franklin is hunched over an X-ray diffraction machine. She is a perfectionist. Her lab notes are meticulous, every data point recorded with painstaking precision. Her colleagues find her difficult, even cold. But the truth is, she was simply focused, uninterested in the social gains of academia. In the 1950s, science was a man's world. Franklin wasn't just fighting to make a discovery. She was fighting for her right to be recognized as an equal. She was studying a mysterious molecule called DNA. Using a groundbreaking technique, she fired X-rays through crystallized DNA fibers. The scattered rays left intricate patterns on photographic film, each one a unique fingerprint of the molecule's internal structure. This was incredibly demanding work. Then, one night in 1952, she captured an image that would alter the course of science. Photo 51. It was a striking, elegant X-shaped pattern. It screamed helix, but Franklin was a cautious scientist. She wanted more proof before making a bold claim. That very rigor, that demand for certainty, would tragically cost her the lead in this race. While Franklin meticulously gathered evidence in London, a very different scene was unfolding in Cambridge. There, a brash 23-year-old American named James Watson had teamed up with Francis Crick, a brilliant and boisterous physicist turned biologist. Their approach was less about patient experimentation and more about inspired guesswork. They built physical models out of wire and cardboard, trying to piece together the puzzle of DNA's shape based on existing chemical knowledge. And they weren't alone. Across the Atlantic, the world-famous chemist Linus Pauling was also closing in on the answer. The race for the secret of life was on. The story takes a controversial turn in the winter of 1951. Watson visited London and met with Maurice Wilkins, Franklin's colleague. Wilkins, who had a tense and difficult relationship with Franklin, showed Watson her data without her knowledge or consent. Most critically, he showed him Photo 51. For Watson, it was a revelation. Seeing that X pattern confirmed his hunch. DNA had to be a helix. Whether Wilkins's act was born of carelessness or a deeper gender bias remains a subject of debate, but for many, it was a theft of intellectual property. When Franklin later learned her data had been used without her permission, she was justifiably furious. Back in Cambridge, armed with the crucial insight from Photo 51, Watson and Crick resumed their model building with renewed fire. They knew DNA had 4 chemical bases, A, T, G, and C, but how did they fit together? Twisting wires and cutting cardboard, they finally stumbled upon the solution in the spring of 1953. It was a double helix, 2 strands intertwined like a spiral staircase. And Watson had another epiphany. If the base adenine always paired with thymine, and guanine always paired with cytosine, the width of the molecule would be perfectly uniform. It was an elegant, beautiful solution. On February 28, 1953, they finished their model. Bursting with excitement, Crick famously walked into a local pub and declared to everyone, "We have found the secret of life." Just a couple of months later, on April 25th, their findings were published in a short 1-page paper in the journal Nature. The paper ended with one of the most significant understatements in scientific history. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In that single sentence, they had explained the very basis of heredity. When Rosalind Franklin read the paper, she must have felt a profound sense of betrayal. Her data, her beautiful Photo 51, had been the key, yet her contribution was barely a footnote. She had been so close. Had she been less cautious, she might have published first. But she was trapped a woman in a man's world, punished for her own high standards. Tragically, Franklin's story doesn't have a happy ending. She continued her brilliant work, producing further X-ray evidence that confirmed the double helix model. But in 1956, she was diagnosed with ovarian cancer, very likely caused by her years of exposure to X-ray radiation. She died on April 16, 1958, at the age of just 37. 4 years later, in 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize for the discovery of DNA structure. Because the prize is not awarded posthumously, Rosalind Franklin's name was left out. Her monumental contribution was almost erased from history. It took decades for historians to piece together the truth and restore her rightful place in this incredible story. The discovery of the double helix didn't just win a Nobel Prize. It launched a revolution. For the first time, humanity understood how life copies itself. DNA wasn't just a chemical. It was a code, a blueprint for building every living thing. This kicked off the entire field of molecular biology. By the 1970s, we were learning to cut and splice DNA, giving birth to genetic engineering. The impacts are everywhere today, in forensic science that solves crimes, in agriculture with genetically modified crops, and in medicine that can detect diseases before they even start. Think about it. In 2003, we completed the Human Genome Project, mapping all 3 billion letters of our genetic code. It took 13 years and cost billions. Today, you can get your entire genome sequenced in hours for less than $1,000. And during the COVID-19 pandemic, the mRNA vaccines, a direct descendant of this DNA research, were developed in under a year, saving millions of lives. All of it traces back to that race in the 1950s. Now, we stand on the brink of another revolution with technologies like CRISPR, which allow us to edit the DNA code directly. This brings the promise of curing genetic diseases and cancer, but it also raises profound ethical questions. Should we edit human genes? Where do we draw the line? The debate rages on. Though she died without the recognition she deserved, Rosalind Franklin's legacy is now celebrated. Her name adorns university buildings and research grants. She is remembered not just for her science, but as a symbol of the struggle against injustice. Her story reminds us that science, like all human endeavors, is not always perfect, but ultimately the truth endures. The discovery of DNA was our journey to the center of ourselves. It showed us that with curiosity, persistence, and reason, no secret, whether in the vastness of space or in the tiny nucleus of a cell, is beyond our reach. Thank you so much for watching. If you found this story as fascinating as I do, please hit that like button and subscribe for more deep dives into the history of science. See you in the next one.